All right, the cell has undergone meiosis 1, and now we're entering into meiosis 2. The spindle forms. This is going to look a lot like mitosis, by the way. Meiosis 2 looks like mitosis. Chromosomes positioned at the metaphase plate again in metaphase 2. Kinetochores of sister chromatids attached to microtubules from opposite poles, just like before. And in anaphase 2, sister chromatids separate towards opposite poles. Now, this is important. In metaphase, or in meiosis 1, chromosomes consisted of um, chromosomes that still had two chromatids each, right? And so they separated in anaphase 1. So if you want to use terminology, it helps to use hand puppets, I think, when you learn this. So in anaphase 1, we saw homologous chromosomes separate. In anaphase 2, sister chromatids separate. Say, and so anaphase 1 was this, anaphase 2 is this. So it's more like mitosis when sister chromatids separate into daughter chromosomes. So sister chromatids, daughter chromosomes, right? Um, Telophase 2, nuclei reforms, chromosomes go back to chromatin, cytokinesis takes place, the cell divides. It's a lot like mitosis. And at the end of this, 4 daughter cells are produced, and they are all genetically distinct from each other. That's really important. 4 daughter cells are produced. First of all, they're haploid. 4 haploid daughter cells are produced, and they're genetically different from one another. This is why meiosis is so important. So if we compare it to mitosis, I apologize for my video being down here in the corner, but let's walk through this. We start with this cell, a 2n of 6. There's 1, 2, 3, 4, 5, 6 chromosomes there. That's a diploid cell of this organism. It undergoes S phase, so those chromosomes copy themselves. The DNA replicates. Here we have 1, 2, 3, 4, 5, 6 replicated chromosomes there. So they're like this. They have chromatids, right? 2 sister chromatids there stuck together. In the case of meiosis, you have the same thing. You have chromatids replicated, but homologous chromosomes are paired up. So once again, a homologous chromosome, if you haven't grasped what that even means yet, remember, we're talking about like your cells. You have 46 total chromosomes in there, but each one has a partner. Like chromosome 1, let's say chromosome 1, you have chromosome 1 through chromosome 23, okay? 1 through 23, and each one has a partner. So there's 2 of each one of those. That's why there's 46 total. One of that pair you got from mom, one of that pair you got from your dad, okay? That's what a homologous chromosome is. Like chromosome number 1, you got one from your mom, one from your dad. There's 23 total pair in your body. So here we can see those pairs are partnered up like this. They're in synapse. They're stuck together, and there's crossing over going on. They're sharing parts. They're swapping DNA, which means when you're done with all this, you have a chromosome that's got most of your mom's DNA on it, but a little bit of your dad's DNA stuck in it too, so you have a new combination of DNA. That's what's going on here. Then we can see in metaphase, in mitosis, everything's lined up in a single column line, basically. And then what's going to happen in anaphase, of course, sister chromatids are going to separate. But in meiosis, everyone's still paired up. Homologous chromosomes are still paired up at the metaphase plate. And in anaphase 1, homologous chromosomes separate. Then in anaphase 2, sister chromatids separate. And we end up then, I want to show you this. This is also very important. At the end of anaphase 1, we have 2, or I should say at the end of telophase 1, we have 2 haploid cells produced. Now they still consist of chromosomes that look like this, right? They're still replicated chromosomes, but there's 3 of them. 1, 2, 3 total. Count the centromeres to count the chromosomes. 1, 2, 3. There's 3 X's in there. Those X's are still 1 chromosome each. They're still considered to be 1 chromosome. So this is 3 chromosomes. This is 3 chromosomes, and we started with the cell with 6 chromosomes. So we have a haploid cell, but we're not done because those chromosomes are still replicated chromosomes or duplicated chromosomes. They need to separate yet. That's what happens in meiosis 2. Those sister chromatids separate, and now we end up with 1, 2, 3, 4 cells produced that are haploid. So they have 3 chromosomes each, unlike the 6 we started with. And they all have different combinations of chromosomes. Different numbers of red versus blue chromosomes, and crossing over has mixed up some of the red and the blue here, the paternal and the maternal traits. You can check out the table in your book that compares these two methods of division. It's kind of important you do that. So variation. This is where meiosis becomes so important, is that not only does it make haploid cells, so if it didn't, think about the consequences we would have. If your sperm and eggs had 46 chromosomes in them, like the rest of your body, then when a sperm met an egg, you would have 92 chromosomes in your offspring. That wouldn't work. That individual would not develop correctly. They probably wouldn't even be born. Well, I know they wouldn't be born. So instead, what we do is we make sperm and egg with 23 chromosomes. We half that number, 23. And when sperm and egg come together, then that offspring has 46, just like you have. So first of all, meiosis makes haploid cells. That's important. But it also mixes up genetic material, leading to variation. And variation is absolutely vital for natural selection to take place. If we were all clones of each other, natural selection wouldn't work. So it gives us new combinations. It's not making new DNA really, but it's mixing up combinations of DNA from your mom and dad, putting them onto the same chromosome. Sources of variation include these three things. The first two are the important ones for this chapter. Crossing over and independent assortment of chromosomes. Mutation will come up a couple chapters from now when we talk about replication and gene expression. So here's a cartoon version of what I'm talking about. Here's dad, here's mom. Okay? Dad has, let's say we're looking at chromosome number 1. Okay, chromosome number 1 has 2 pairs. Dad has 2 of them, mom has 2 of them. I made those the same color to avoid confusion. Say dad has 2, mom has 2. Okay? They each have homologous chromosomes for chromosome number 1. But when they make a sperm or an egg, only one of those of that pair ends up in the sperm or the egg. Then that sperm and egg fuse and we have this daughter produced. She has dad's blue chromosome and mom's red chromosome. Now when she goes to make an egg, only one of those 2 chromosomes ends up in that egg. And it has the possibility of having a piece of the other chromosome stuck to it because of crossing over. This is where that genetic diversity comes from. We can mix and match because of crossing over. So that's crossing over. That's what crossing over does. It takes a piece of this chromosome here and sticks it to a piece of this chromosome here. Okay? That's when this daughter goes to make an egg. Now, the next level of variation that I want to talk about. Your book will talk about also the fact that like fertilization is a chance for variation because you'll have like 10 million sperm in one egg and only one of those sperm fertilizes the egg. So there's another source of variation you might read about, and I should mention that. But this is the one that we're going to focus on in lecture, and that is independent assortment. So if we go back in time to metaphase 1, when those chromosomes line up on the metaphase plate, okay? So pretend my left hand is maternal and my right hand is paternal, and we're just looking at chromosome 1. They could line up like this, and there's an equal chance they'll line up like this. That's independent assortment. It's independent. They could go either way, how they line up at the metaphase plate, okay? That might not seem like a big deal, but it really, really is, because every pair of chromosomes has that option of lining up either way. And so you have 23 pair of chromosomes. So those are a lot of combinations that they could line up as. All the blue ones, all the maternal ones could, or all the paternal ones could be on the left, and all the maternal ones could be on the right, or vice versa, or any combination thereof. So we're going to tone it down from the human example and look at this example here, an organism with a diploid number of 4. 1, 2, 3, 4, and see how they could line up. So possibility 1, you can see on the left, all the blue are on the left, all the red are on the right. Possibility 2, there's a blue on the left for the top pair, and there's a red on the left for the bottom pair. That's what independent assortment looks like. And the important thing is that results in different combinations. We're not even worrying about crossing over here. Let me point that out. We're not worrying about crossing over at all in this figure. We're going to pretend it doesn't exist for now. And we're just going to look at independent assortment, okay? So thanks to independent assortment, there'd be 4 gametes produced. And in this case, 2 would be all blue and 2 would be all red. And in this case, you would have a combination of blue and red chromosomes in each gamete because of independent assortment. And that's just looking at an organism with 4 chromosomes. You have 26 pair or 23 pair of chromosomes in your cells. 23 pair. Each one can line up either way. So even without crossing over, you have over 8 million possibilities in your gametes. So here's another example with a cell of 2n of 6. All the different ways those chromosomes could line up. And the way you can calculate all the ways they could line up, all the combinations they could produce, you take 2 to the haploid power. 2 to the power of the haploid number. So for this cell, the diploid number is 1, 2, 3, 4, 5, 6. So the haploid number is 3. So 2 to the third is how many possible combinations there are, and that's 8. And you can see here there are 8 different ways these chromosomes could line up. I'll let you look at that and study that. In human beings, it's 2 to the 23rd possible combinations. That's over 8 million, and that's not counting crossing over. So you can have more kids than with the same partner as there have ever been humans on the planet, and none of them will come out as identical twins if they're born at, if they're all made by separate fertilization events. Pretty amazing. All right. So that's where variation comes from. I have a couple of questions here. I'm just going to scroll through these, and you can pause the video and see if you can answer these. At what stage is the independent assortment of chromosomes determined? There's a question for you to try to answer. Oh, you might be able to find the answer on this slide. All right. So independent assortment takes place during metaphase 1. Okay. All right. The last thing I want to mention before we go is we're going to have a couple more slides on errors. So sometimes errors take place in meiosis, and this is called, or mitosis actually, either one. These are called non-disjunction events. That's when chromosomes, instead of doing this, might all go in one direction, or instead of this, it all goes in one direction. Failure of some or all chromosomes to move to opposite poles during a meiotic division, and it could be mitosis too. I want to point that out. But it usually has bigger consequences if it happens in meiosis. Why? Because if you have a mutation, an error in a cell of your little finger, where you have too many chromosomes in one of those cells, that might not be a big deal. Okay? It probably won't be a big deal. But if it happens in an egg or a sperm, and then that egg or sperm gets fertilized, right, or there's a fertilization event, then every cell in that offspring's body has that mutation. So if this happens in a gamete, then it's a serious consequence. This is how you end up with something like Down syndrome, because you have one extra chromosome in that egg or in that sperm, and then fertilization happens, and then every cell in that individual's body has that mutation. So if a mutation occurs in the gamete, it's found in every single cell that gamete gave rise to. So here's an example of that. Down syndrome is known as trisomy 21 because here's those 23 pair of chromosomes, and the 21st chromosome has 1, 2, 3 copies. And because of that extra gene expression, development is changed and Down syndrome symptoms develop. So that's known as non-disjunction. That's how that occurs. Okay. What are 3 ways in which gametes from one diploid cell can be different from one another? There's a question for you. In this cell, what phase is represented? Once again, you can go back, hit pause, and try to answer these. Consider an organism of karyotype 2n equals 4. Which diagram depicts metaphase 1? Which one of those is metaphase 1? With the organism 2n of 4. I'll let you determine that. All right. And lastly, consider an organism karyotype. Karyotype just means a picture of the chromosomes. 2n of 4. Which diagram depicts metaphase 2? All right, see if you can answer those, and have a good one.